All right. So, good afternoon to everyone. Thanks for being around for this new week, this new talk that we're gonna have today. I'm gonna briefly introduce Djalma de Souza Lima Júnior. Djalma graduated in biology at Federal University of Ouro Preto. He did his master's and PhD at the USP Ribeirão, and then went to work at NIH. Did a lot of stuff there, right? With a lot of important people, right, Djalma? I'm gonna leave that to you, so you can explain to people. But yeah, you spent a long time in the U.S. and now you're returning back as a young researcher with a fellowship by FAPESP, rejoining the School of Medicine of Ribeirão Preto. And I hope, I wish you get a position soon, Djalma. For sure, you deserve it. And in a few months or maybe years, you'll be hired, tenured there at Ribeirão Preto or wherever you choose. Okay. So thank you very much for being around, and please feel free to present your talk. Okay. Thanks a lot.
Okay, great. Thank you. So first of all, I'd like to thank these webinar organizers for inviting me to give this talk here today, especially the Djalma to invite me here. So I'm really happy to give this talk today. So thank you very much. And as Thiago said, I did a postdoc at NIH in the Yasmin Blockades lab. So it was a great experience, like for 6 years, around 5 years and a half. And now I'm back here and start to a new phase of my career at Ribeirão Preto Medical School as a young investigator, or young research. And today I'm going to talk a little bit about what we did during my postdoc at NIH, and also a little bit about what we are doing now, and the focus of my lab and the focus of my project. So, and just to start the talk here. So today I'm going to talk a little bit about the endogenous retrovirus control of the host skin immunity. So we're going to focus on skin immunity and endogenous retrovirus, right? Like a endogenous microbiome or microbiota, or to be like better, endogenous virome. And just to introduce a little bit about the microbiota. So we know that the microbiota can modulate the physiology of different tissues, such as the skin, the gut, the lung, and, for example, the liver. And the microbiota is directly involved at steady state. For example, it's directly involved in the homeostasis of the epithelial barriers, in the education of the immune system, and also the microbiota can promote, for example, the host resistance to pathogenic microorganisms. However, exogenous factors, for example, stress, antibiotic usage, aging, or diet, for example, diet change, it can, just to minimize here, it can cause a dysbiosis, can change the microbiota composition in different tissues, which can lead to inflammatory disease, such as allergies, autoimmune disease, or metabolic syndromes and inflammation. However, the mechanism by which the microbiota control the host physiology or control inflammatory processes in different tissues, it's remaining clear. So in this context, we decide to investigate how the microbiota could influence the skin immunity. What's the mechanism by which the microbiota influence or impact on the skin immunity? And then to analyze that, the Gut Lab established a model in the lab using a skin convention, the Staphylococcus epidermidis. That is a convention highly present in the human skin. And interestingly, this microbe can colonize the mouse skin surface. So as you can see here, using a model when we gently apply apply the Staphylococcus epidermidis in the ear, we observe microbe colonization in the mouse skin surface. For example, here at different time points, for example, 14 days, 30 days, 60 days, or for example, 6 months, we have a decrease, but we continue having a microbe colonization in the skin. And also, we observe that this colonization promotes the accumulation of T cells in the ear or in the mouse skin, for especially CD4, CD8, and the mate cells. And as you can see here, we have a massive accumulation of CD4 and CD8 T cells, and especially CD8 T cells that are making IL-17 and interferon gamma, for example. And also, we have an increased or massive accumulation of innate cells, innate like lymphocytes, making IL-17 cells. However, one interesting point I'd like to highlight here is that despite this massive accumulation of T cells in the skin, we do not have skin inflammation. We do not have a skin pathology. As you can see here, after staph IP colonization, we did observe skin inflammation. For example, if you compare the topical application, the gentle application of staph IP with an intradermal injection, we observe in an intradermal injection a huge increase of neutrophils and monocytes in the skin, which is indicative of skin inflammation. However, after the topical application, we didn't see any difference in these cells infiltration in the skin, indicating that the topical application induced a massive accumulation of the T cells or immune cells in the skin, but we do not have a signals of inflammatory process, which we call that homeostatic immunity. So this kind of immunity is called homeostatic immunity. So at steady state, the topical application, the Staphylococcus epidermidis colonization in the skin, it's caused homeostatic immunity. It's promote homeostatic immunity, but not an inflammatory process. Okay. And another work in the lab to try to understand how this massive accumulation is induced in the skin, it's showing that despite of the inflammatory process, we don't have inflammatory process in the skin, but we observe one interesting point that is a huge increase, a strong increase, or a strong induction of antiviral response in the skin. So as you can see here, keratinocytes isolate from the skin. Here was a tissue skin. So that when we analyze, when we perform a bulk RNA sequence of the skin tissue after the topical, the Staphylococcus epidermidis staph IP colonization, we observe in this tissue a strong antiviral immune response. So it's a paradoxical response because we have here a colonization with a no-cause microbe that doesn't cause any inflammation, but it can induce a strong antiviral defense program. And looking at the literature, it's at the beginning it's perplexing, but when you look at when you take a look in the literature, we realize that it was not a big novelty, a big surprise, because other studies published, for example, by the David Art's lab or the Akiko's lab, they also show that the microbiota, especially the gut microbiota, has the ability to promote a strong antiviral response in different tissues, for example, especially in the lungs. However, the mechanism by which this microbiota, or the mechanism that link the microbiota and this strong antiviral signature is completely understood, it's completely unknown. So it leads us to revisit a fundamental biological concepts about our biology, that of course we are composed of exogenous microbes, for example, bacteria, fungi, and virus, but also despite this exogenous microbiome, we also harbor endogenous elements such as heterotransposon or hetero elements that have been also to control the physiology of the pathological process in different tissues, including tissue homeostasis and tissue inflammation. So about the endogenous heteroelement and this heterotransposon, we know that 50% of our genome is composed by this sequence that is divided in different classes, in different types, for example, line, sign, and ERVs. And regarding the ERVs that are our focus here in this talk, the ERVs compose around 10% of the human and the mouse genome. And similar to other heterovirus, the ERVs also replicate via the reverse transcription or via the reverse transcriptase activity. And this process is critical for the virus replication. So similar to other retrovirus, for example, HIV or herpes virus. And then in this context, we decided to investigate what are the mechanisms by which the microbiota can trigger the host immune response in different conditions, for example, steady state and inflammatory settings, in a context, for example, of ERVs reactivation. So initially, we assessed the skin signature in keratinocytes after the Staph colonization, the skin microbial colonization. And then to address this point, we colonize the mice with Staph, and then we sort keratinocytes after 7 days post-colonization. And then we analyze the gene profile by bulk RNA sequence. And as you can see here, similar to previous data showed here, we observe that the Staph colonization, it's induced overall positive regulation of inflammatory response, especially the induction of type 1 and type 2 interferon signaling pathways. And also we observe higher levels of genes implicate in an antiviral immune response. So this data indicate that the colonization, the Staph colonization, it's induced a broad transcriptional program in keratinocytes, including increase of a type 1 interferon response pathways. And then next we analyze what's the impact of the type 1 production, the type 1 interferon in a homeostatic immunity induced by the Staphylococcus epidermidis colonization. And then to address this point, we treat the mice with anti-FMR, with a blocking antibody against type 1 interferon receptor. And then we colonize the mice with Staph, and we analyze the T cells infiltration, the T cells response accumulation in the skin. And as you can see here, we observe that the anti-FMR, the FMR blocking, or the blocking of the type 1 interferon receptor, it's caused a strong, a drastic reduction of T cells accumulation in the skin during the Staphylococcus epidermidis colonization. It includes TC17, TC1, Th17, Th1, mate cells, and the gamma delta T cells making IL-17. So you have here a broad reduction of T cells in the skin after the type 1 interferon receptor signaling blocking. So this data indicate that the type 1 interferon response, it plays a crucial role in the control of the immune response to cutaneous conventions or to cutaneous colonization with skin microbes. So next we analyze what are the mechanisms by which the Staph promote this cutaneous T cell response in a context of antiviral immune response. And then when we analyze the ERVs expression or the endogenous retrovirus expression in keratinocytes after Staph colonization. And interestingly, we observe that the Staph promote a significant increase of ERVs or of heteroelements expression in the skin. And also, we observe that these heteroelements, they were active because if you look at here, we also have a huge increase, a significant increase of genes involved in the protease and the reverse transcriptase activity, for example, the PRO and the PUL genes. So Staph induced an increased expression of these genes, indicating that these elements, they are active. And in this context, we decide to block the ARV's replication. So to do that, we use a antiretroviral cocktail. Here we use two different reverse transcriptase inhibitors, the tenofovir and the emtricitabine, to block the reverse transcriptase activity, which we're going to indirectly block the ARV's replication in the host. And using this approach, and associating the mice with staph-IP, we observe that the antiretroviral treatment induced a strong or significant reduction of T cells accumulation in the skin, including CD4 and CD8 cells. So as you can see here, you have a drastic reduction of T cells accumulation in antiretroviral treated mice compared with untreated mice. So next we analyze if this treatment, if the reverse transcriptase inhibition, it could also impact on the keratinocytes response. And to address this question, we treat the mice with the antiretroviral, and then we associate these mice with staph-IP, and we sort keratinocytes, and we perform a single cell RNA sequence to look at the gene profile, the transcriptome of the keratinocytes after the antiretroviral treatment. And in this single cell, we identify different clusters, in this case here, 18 different clusters, that was divided in 5 different keratinocyte subsets, okay? And when you look for the gene expression, we observe that the antiretroviral treatment, it's significantly reduced the expression of different genes involving different physiological processes in keratinocytes. For example, genes involved in the antiviral defense, genes involved in the antigen presentation, and also genes involved in wound healing. And to validate this single cell RNA sequence, we check the impact of the antiretroviral sequence, the antiretroviral treatment in a context of the wound healing promoted by the Staphylococcus epidermidis colonization. And to address this point, we colonize these mice, the black 6 mice with the staph-IP, and then we treat the mice with antiretroviral, and we analyze the ability for the bacteria to induce the tissue repair in these mice. And as you can see here, when we treat the mice with Staphylococcus epidermidis, we observe a reduced epidermal tone, shown here by the keratinocytes staining. Here we use a keratin 14, it's a keratinocytes marker, to look at the keratinocyte, the epidermal tone of the keratinocytes proliferation. And we observe that the antiretroviral treatment reduced the epidermal tone, that is indirect readout for the wound healing or the tissue repair, compared, for example, with mice treated with a vehicle. So this data indicating that in line with the reduced keratinocytes and T cell response that is directly involved in the tissue repair, the antiretroviral treatment impairs the ability of the Staphylococcus epidermidis, the staph-IP, to promote the wound healing. And the next, we investigate what are the ARV's downstream mechanisms involved in this context, or the downstream signaling mechanisms involved in the ARV's reactivation during the staph-IP colonization. So we know that the retrovirus, once activated, they can produce a DNA that can accumulate in the cytosol compartment, in the intercellular compartment, in this case here in the cytosol. And once accumulated in the cytosol, this material, this antigen, this can be sensed by different receptors in the cytosol. For example, the cGAS receptor, that is it's a innate sensor able to recognize DNA. And once activate the cGAS, induce the STING activation that will lead to if IF3 phosphorylation and lead to type 1 interferon response or type 1 cytokine secretion. So in this context, we investigate if the cGAS STING pathway could be involved in a homeostatic immunity promoted by the STFAP or the Staphylococcus epidermidis colonization. And then we associate, we colonize wild type mice and knockout mice to cGAS and STING. And we observe that deficient mice to this pathway showed a huge decrease, a significant decrease of T cells accumulation in the skin, similar to anti-retroviral treatment. And also, for our surprise, when we delete the STING signaling only in keratinocytes, we observe a drastic reduction of T cells accumulation in the skin during the STFAP colonization. So taken together, this data showing that the STFAP-induced skin or homeostatic immunity, it's dependent on the cGAS STING signaling in keratinocytes. So as a conclusion of this first part, we observe that the skin microbe colonization, in this context here, in this case here, the Staphylococcus epidermidis colonization, but we also observe similar results in after the colonization with Staphylococcus zylosus, Staphylococcus aureus. So in this case, we can say the skin commensal colonization, it can trigger a discrete ERVs expression that via cGAS STING pathway, it's promote a skin homeostatic immunity. Okay, but we also know that the microbe colonization can promote inflammation. So it can promote homeostasis in different tissues, but it can also promote in different contexts. It can also promote tissue inflammation. So we next evaluate if in a context of inflammatory process induced by skin commensal colonization, the ARVs can also control the ability of the microbiota, the skin microbiota, in promote the skin inflammation. So to address this context, we use a high-fat diet because we know that the high-fat diet, it can cause a skin or it can cause a microbiota dysbiosis, which is correlated with, for example, skin disease exacerbation, or such as, for example, atopic dermatitis and psoriasis. So there are several reports showing that the microbiota or the dysbiosis or this imbalance in the microbiota caused by the dietary change, it can promote, it can exacerbate, for example, different diseases, including skin disease. So in this case, so we use this approach. We use the dietary change to evaluate if in an inflammatory context, the microbiota could impact the skin inflammation through the skin microbes colonization in a context of ERVs expression. So to address this question, we observe that the STFAP colonization at steady state, it doesn't cause any inflammation. So as we can see here, for example, when you compare unassociated mice with the STFAP-associated mice, we didn't observe any difference in the skin thickness, that is indicative of skin pathology, between these two groups. But one interesting point that when you place the mice or when you, with a high-fat diet, or when you fed the mice with a high-fat diet, we observe that the same colonization, the STFAP colonization of this skin, it's cause a skin inflammation in a context of high-fat diet. So mice fed a high-fat diet showed a huge increase of the ear thickness, which is indicative of skin pathology. And when we observe here, we have an increase in the cell, immune cells infiltration, increase in the skin pathology, and also a huge increase of the keratinocytes layer or keratinocytes proliferation in mice colonized with STFAP in a context of a high-fat diet feeding, showing that when you change the diet of the mice, you can, instead to promote the homeostatic immunity, you can promote tissue inflammation after the skin colonization. And for our surprise, when we fed the mice with a high-fat diet without microbe colonization, we observe a huge increase, a significant increase of the ARVs expression in the skin. But when we associate these mice with STFAP, we have an aberrant ARVs expression in keratinocytes isolated from these mice. So showing that my skin microbe colonization together with a high-fat diet or dietary change, it can promote aberrant ARVs expression in the skin. And also using the same approach to observe the impact of the reverse transcriptase inhibition, when we treat the mice fed a high-fat diet and colonized with STFAP, when we treat these mice with anti-retroviral drugs, we found a significant reduction of the ear thickness, a significant reduction of the skin pathology caused by the conventional colonization. And also we observe a drastic reduction of T cells accumulation in the skin. Okay. So in this conclusion of this work, we observe that at steady state, the skin microbes, it's promote the ARVs, a discrete ARVs expression that can be sensed by the STING signaling pathway, promoting the homeostatic immunity. However, when you change the diet in a context of dietary change, this same microbe, it can cause a huge increase, a strong increase of ARVs expression, which will promote tissue inflammation and tissue pathology. Okay, and as a follow-up of these studies, we also know that the ARVs are associated with different inflammatory disorders. So there are many reports in the literature showing that the ARVs is associated. It's just a correlative study, so we don't know what's the exact impact of the ARVs in this context, but there are many reports showing that for example, in a context of different inflammatory disorders, for example, multiple sclerosis, lupus, diabetes, cancer, we have a strong ARVs expression, and it is correlated with the disease severity. So more severe disease will have, in general, have more ARVs expression. But the real impact, the real impact of the ARVs in a context of this disease, in a context of inflammatory disorders, it's remain unclear. So in this context, as a follow-up of this previous study, we decided to investigate what's the impact of the ARVs in different inflammatory disorders. And here, in my previous work, investigating a public gene bank, a public RNA sequence bank in different diseases, we also observed that the human psoriatic lesions express a huge amount, or express increased there is an increased ARVs expression in these lesions. And to confirm this data, we use an experimental model of psoriasis using a imicimod application. So you apply the imicimod in the ear skin, and evaluating the ARVs by flow cytometer. Here we use an antibody that's specific for a protein, envelope protein expressed by one specific family of ARVs. But using this approach, we observe that after the imicimod application, or which mimics the psoriasis in human, we observe a strong ARVs expression in skin cells, skin immune cells. You can consider here, in this case, for example, the lung hair cells and keratinocytes, that also are important cells responsive in the skin that contribute to initiate the immune response of the skin. So we observe a strong ERVs expression in these cells in the skin after the MIC mod application. And when we block the ERVs replication, when we block the reverse transcription in this context, we also observe a decrease in a skin inflammation, the skin thickness, and also in a skin pathology. For example, if you compare here, the skin pathology in the mice that we apply the MIC mod, or the mice we've treated with antiretroviral untreated mice, we have a strong reduction in the pathology in the ear and a significant reduction of the ear thickness. And looking for the T cells accumulation in this tissue, we also observe that the antiretroviral treatment drastically reduced the Th17 response or TC17 response. That is the major response involved in an inflammatory process in a context of psoriasis. So we observe a Th17 reduction and also a TC17 reduction after the antiretroviral treatment. And another interesting data that we got in this experiment is that the MIC mod applications can also cause a local immune response, a skin immune response, but it can also cause a systemic immune response, especially T cells response or immune response in the spleen. So when you look at the spleen of the mice treated with antiretroviral, we observe here a huge decrease in the size of this spleen. It's like a macrophenotype, and also a significant decrease in the spleen weight compared, for example, with the mice treated with the vehicle. So, and corroborated with this data, we also observe a strong reduction of the type 3 immunity in the spleen. For example, a Th17 and TC17 cells accumulation or response in the spleen, indicating that the ARVs are directly involved in the psoriasis disease, are directly involved in the psoriasis inflammation in the skin. So the ARVs expression has the ability to also control the development of psoriasis disease or psoriatic lesions. So I'd like to finish here. I'd like to thank the Yasmin Belcage for all the support she gave me during my postdoc in her lab during the past 6 years, and all the people involved, the Belcage lab member involved in the development of this project, Sid, Verena, and Nicholas Bolador, Nicholas Collins, and also the Santi that is here. So it's a big pleasure. It was a big pleasure to work with you, Santi. And also the lab members and the external collaborators at NIH in different institutes, NCI, LPVD, NCATS, and also the George Cassiotti from Francis Crick Institute. And I'd like to thank the financial support from NIH, from PIL, from FAPESP. And finally, I would like to mention here that I'm hiring now PhD students and master degree students and undergraduates. So who wants to join this amazing lab, the Laboratory of Endogenous Viral and Barrier Immunity, please don't hesitate in contacting me to schedule a time to discuss a little bit more about the projects, the lab focus, and also to discuss a little bit in details about the selection process of the basic and applied immunology program at Ribeirão Preto Medical School. So here it's a picture of the part of the São Paulo University campus in Ribeirão Preto, and my lab is here in a central building. That's a wonderful build. It's a central building at Ribeirão Preto Medical School. It's a wonderful build. So who wants to join my group, please contact me and we can talk a little bit more about these positions. Okay, so thank you very much for attention, and I'm happy to take any questions.
Nice, Djalma, very nice work. Yes, so I would like to ask for the students for questions. for Djalma. Are you actually sitting at the old Diego's table, or is it in the same lab? Are you staying in the same lab?
Not exactly. It's part of the Diego's. It's the same because I'm using like his old lab as well, but I'm using another part as well. I'm using the of course, you know, the Cellulops lab, like the old Cellulops lab. So I'm using just two labs, the Diego's old lab and the Cellulose old lab.
Did you allow him to do that, Diego?
No, exactly.
I think
no, he's not working at the department anymore. So the department chief like allowed me to use there. It was a big negotiation, but it was fine.
I think he's much better than I was when I was there, right? Because he's got Cellulose lab. The whole Cellulose lab for him, so it's much better.
Exactly, because the Cellulose lab is completely equipped. So in this case, I could save some money. Yeah. So now it was much better. Yeah, I have different equipment there. So for me, it was wonderful. Yeah.
Okay, we have a question by Luisa. Luisa, please.
Yeah.
Hey, Djalma, very nice talk. I have a question. It's more regarding the biology of the bacteria, because I don't understand a lot about Staphylo, but I was thinking about this wound healing properties that it has. And when you have like a strong T-helper 17 response, as happened in psoriasis, it's like enhanced. But do you know about the bacteria, if it can produce some kind of metabolite like in steady state, besides this enhancing of the IL-17 response and other stuff that it has that promotes the wound healing, like some kind of metabolite that is produced by Staphylo that can help in this context, and it's like impaired when you have like the antiretroviral association or treatment of these mice?
Yeah, it's a great question. So far, right? We just analyzed the bacterial colonization, but I guess we don't know which metabolite, for example, from Staph IP or from other bacteria can promote like the activation. But what we know so far is that the toll-like receptor signaling, which is going to recognize some antigen from the bacteria or some metabolites, I don't know. But we know that the toll-like receptor, for example, in this context here, in a Staph IP context, that it's binding the major toll-like receptors involved in the Staph IP response is toll-like receptor 2. So I didn't show here, but when we deplete, for example, when we use knockout mice to toll-like receptor 2, we observe a decrease in the ARV's expression in a context of Staph IP association. So you know that toll-like receptor 2 is important for ARV's expression. We also know that NF-kappa B, it's other reports showing that, but we know that NF-kappa B activation is important for ERV's expression. So that's one transcription factor involved. We have other transcription factors, but the NF-kappa B is the major transcription factor involved in the ERV's activation. And toll-like receptors activate NF-kappa B. So I think that, for example, in the context of Staph IP or in Mickey mod application, because the Mickey mod also activate toll-like receptor, for example, 7 and 8, the NF-kappa B activation is like the crucial or the key molecule involved in the ARV's expression. But exactly which metabolite or which antigen from bacteria that induce that, we don't know in this case. Yeah. But that is one, just to complement the question, it's one thing that we're gonna start to test now, not in a context of ERVs, because just to, I didn't say that, but now I'm focused to understand the ERV's impact on type 2 immunity using different models, including the helminths infection. So, and then in this case, in a helminths infection, we're gonna start to work with the vesicles, secret by these helminths, for example, by the helminths, to investigate. We know that the helminths, it's induced the ERVs, but we don't know like how it's induced. But we believe that the vesicles, for example, secreted by the helminths, it can activate different sensors, whatever, that can lead to ARVs activation. And we're gonna check which molecules in these vesicles is involved in the to engage. We can say that, like, to engage the immune system in a context of ARVs expression. Yeah.
Great. So, Diego, we have a couple of questions in chat. Could I go with them first? Okay. So,
yeah,
Douglas, do you want to ask your question? Douglas, are you there?
Or wait.
I'm not seeing him anymore.
Maybe just read it.
Yeah, I'm gonna read it here.
Yeah, there are some. I opened the chat here. Yeah. Can you read or? Yeah, if you want, I can read here, then I can respond. Okay. I have opened the chat here. Yeah.
Yeah.
You can read the
Great
chat.
talk,
Yeah.
Djalma. You have shown that during colonization, there's an increase in interferon response. Did you try to infect those mice with virus and see if they're protected from the viral infection?
No, no. In this case, no. We didn't check, for example, in a context of infection disease, for example, virus infection or other infection. What's the role of the skin conventional colonization, the ARVs expression? What we know is that the virus infection, for example, COVID-19, influenza virus infection, they can also promote the ARVs expression in the tissues, for example, in the lung or in the gut, in the case of different via virus infections. So, but for example, if you colonize the mice that gonna promote this type 1 interferon response, and if it can protect the host from a virus infection, we didn't check that. Yeah.
All right. So we have another question here from Lucas. Any clues about the mechanisms by which microbiota is inducing the activation of the retro elements?
Yeah, it's a good point. Just to recapitulate here what I said to Luisa, we believe that the ARVs expression in this context, in microbiota colonization, it's dependent on the toll-like receptor signal and also NF-kappa B activation. Yeah, that's our hypothesis.
Okay, and the last question from the chat. Congratulations, Professor. I have one question for you. Do you pretend to expand the use of endogenous onto retrovirus in another microbiome like guts? Yeah, we intend. So now, as I said, we're going to start some studies involve the helminth infection, and one of these helminths is a H. polygyrus. That's a gut helminth. So it's a specific helminth in the small intestine. So, and then we intend to start some studies to look at the impact of the ARVs in an infection disease, in this context, in a helminth infection in the gut as well. So now we're going to work in a different tissues, lung, gut, and tissue, using different models, for example, allergic asthma, atopic dermatitis, and also a gut infection with helminths, to look at the impact of the ARVs the ARVs expressed in these different contexts. All right. Diego, and then Medium. Go ahead, Diego.
Okay, so very nice work, Djalma. And I have another question. I remember you presenting this in the immunology program workshop, but I have another question also. You characterize very well how this colonization by staph epidermidis induces the expression of this endogenous retrovirus in the keratinocytes and the epithelial cells. Did you check in immune cells if that activates endogenous retrovirus expression in immune cells, and if that can change as well the function of those cells? Yeah, we have done it. It's not my work, just a spoiler. It's coming soon that we're going to publish, like, I suppose in 1 or 2 months. It's already accepted. But in this paper, we're going to show that the ARVs is also expressed by lung cells in a context of staph IP, for example. And it's the lung cells, the ARVs expressed, for example, just as a spoiler, it's already in the bioarchive, so I can say that. So
because we have a differential expression of ARVs in different tissues. For example, at baseline, the ARVs is differentially expressed in different tissues and also in different cells. So in this paper, we just focus on, in my previous paper, we just focus on epithelial cells, the keratinocytes, but now in this paper, we focus on the immune cells. And then when you compare, for example, the lung cells, conventional disease and macrophage, we observe in the context of skin, okay? We observe that the lung cells has a higher expression, the baseline is higher compared with other tissues. And also we observe that the lung cells, we use a different approach. In this case, we didn't use step IP, but we use a detergent application. So because the stress or detergent, the stress caused by a detergent in the skin, it can also trigger ARVs. But using this model, we use a twin and twin and BHI, anyway, a twin detergent to trigger the ARVs. And in this context, we observe that the lung cells is the major cells expressing ARVs involved in the skin inflammation, the skin immune response. So the immune cells is also can also express ARVs and can also be important to skin inflammation and skin response in a context of ARVs expression.
Just a follow-up curiosity, actually. So is this detergent triggering due to damps or is something?
Yeah, we don't know. Yeah, we don't know exactly what happened because in this model we use a BHI that is a mix of it's like a protein soap. It's a mix of different proteins. It's a BHI conjugated with twin. For example, just BHI or just twin doesn't induce ARVs, doesn't cause skin inflammation. But for our surprise, when you combine the BHI plus twin, we have a skin inflammation, and also we have an aberrant ARVs expression. So we don't know exactly what is inducing the ARVs in this contest, but what we know that maybe the detergent, it can break the skin barrier, and then it can. That's the idea, but we don't know. And then it can help the BHI or the antigens present in BHI to assess the immune system, engage in the immune system. We don't know. But what we know is that it's or the stress caused by this, for example, because we know that, for example, the soap, it can, if you use different detergents, it can cause, for example, the skin stress or the skin not lesion, but the skin inflammation, different kind of detergents. And then we are just showing that this type of inflammatory process caused by this detergent, it's mediated by the ARVs and the ARVs expression in lung cells. That's the major message of this work. Yeah.
Nice.
Yeah, it's crazy. Yeah.
Media.
Hello. Hi, Djalma. Thank you very much for your talk. Beautiful, and it was a nice follow-up to our talk from last week as well. We have a couple of questions here, so just take the chance and ask mine first. But I wonder, have you tried starting the retroviral treatment later? Like how late can you start to still have a reduction in the disease, especially in the psoriasis-like model? And if you and have you tried treat the mouse, wait for it to maybe heal a little bit and treat it again? Because sometimes psoriasis, you have those peaks and you have recurrent lesions. And does it work every time, or with time you have different antigens and would be just something on the onset of the disease?
Yeah, I didn't check it yet. When we block in a context of psoriasis, when you block, for example, the ARVs and the effect, for example, it can be a late or can be early during the early response. But in a context of step IP, we block, for example, after decolonization. And then when you block after decolonization, for example, 14 days post-colonization, that's the final time point we use to collect the tissue and to analyze the response. For example, if I start the treatment at 10 days post-colonization, we did not observe decrease, for example, in the T cells accumulation in the skin. So we need to start the treatment at the early stage. So in this case, as I in my data here, we start, for example, 1 week before we start the treatment, 1 week before we start the colonization, because I guess that the innate response induced by the ARVs is responsible for the late adaptive response. Okay. But psoriasis, that's one good point, because we're gonna test, for example, in psoriasis, because psoriasis we know that there are some waves, right? So it's not a constant process. So maybe we can change these waves, or can change the severity of the psoriasis, for example, a chronic psoriasis or not, depend on the ARVs or the antiretroviral treatment. But psoriasis, I just treat 1 day before the MIC mod, for example, application. So I don't know if, for example, if I treat the mice in a late stage, it can also reduce the skin pathology or not. If it has any effect, yeah, like a therapy. But it's one M in these steps. Yeah.
Thank you. And Lucas Bolini has a question. Thank you, Djalma.
Okay. Thank you.
Hi. Thank you for your talk. I have actually 2 questions. I think the first one is, because I think it's quite clear to me that the antiretroviral treatment, of course, changes the gene expression profile in the skin, and all of that. But we understand that the microbiota has a very important crosstalk with the skin, in the sense that as much as the microbiota helps to regulate the homeostasis of the skin, the state of the skin also helps to, and the skin resident leukocytes help to maintain a healthy microbiota in the skin. So my question is, how does this antiretroviral treatment also affect the composition of the microbiota of the skin? This is the first question. And my second question is regarding the type of inflammation that you see in the skin. Of course, the treatment reduces type 3 inflammation, but is it accompanied by an increase in type 2 or type 1 inflammation, or is it more like anti-inflammatory state with more IL-10, TGF-beta production? So how does the inflammation actually change in your model?
Okay, so regarding the first question, both questions are great question. It's a great point. Yeah, I didn't show here, but the effect of the antiretroviral treatment, or the effect of these antiretroviral drugs in the skin, in the staph, for example, particularly in the staph. So we treat just the bacteria with this antiretroviral cocktail, just to check if there was any effect of this treatment directly in the bacteria. And we didn't see any difference, for example, in the bacterial growth in vitro, or something in the bacterial fitness. So in this case, it's specific for the mammal host, not for the bacteria. And when we treat the mice with the antiretroviral and look for the staph colonization, we have a big increase of the CFU, for example, in the skin after the antiretroviral treatment. So the antiretroviral treatments promote the bacterial growth in the skin. That's the message. We believe that as we reduce the host immunity to this bacteria, to the microbiota, because we reduce the homeostatic immunity, it can promote the bacterial growth. Because when you're looking for the genes expression in the keratinocytes, the antiretroviral treatment also reduce the antimicrobial peptides in the keratinocytes. So which is directly involved in the control of the bacterial growth. So when you treat the mice with retroviral, we have an increase in the bacterial growth in the skin, like 2 logs more. It's a very big increase in this case. And about your second question, it's the second question is, sorry, I forgot it. It's
about the type of inflammation, because you see a reduction of type 3 inflammation.
We don't know exactly. We know that it's reduced the antiviral response. So, for example, when you treat the mice with the antiretroviral, but we didn't check, for example, if reduce, for example, cytokines like IL-4, IL-6, TGF-beta, or cytokines that going to promote a different T cells subsets in the context of FMRP. But I have another work that I hope to be published soon, investigating the ARVs on the allergic asthma. So, or in the context of type 2 immunity. So, in the context of allergic asthma, when you treat the mice with antiretroviral, we observe a decrease in the IL-4, IL-13, for example, production in epithelial cells and in dendritic cells. So I guess that the antiretroviral treatment can affect, for example, the innate cells response or innate cytokines. For example, the cytokines that gonna be involved in the T cells polarization. For example, IL-4, IL-6, IL-12, for example. It depends on the context. But in the context of type 2 immunity, allergic asthma, for example, we have a decrease in the chemokines, for example, also. And cytokine secretion IL-4, IL-13, IL-5, for example, in disease or in epithelial cells. So that's what I can tell you about it like so far.
Okay, thank you.
Okay, thanks.
Hi, Djalma. Thank you for the great. I actually have a kind of follow-up question. I don't know if you ever done something on the ERVs in germ-free mice, and how the response in the unexposed skin and the exposed skin occurs in these germ-free mice. And if you didn't do, if you would care to speculate a little bit of what's having normal microbiota would affect these response.
Yeah, that's a great question. So in terms of the ARVs expression in the skin in germ-free mice, we didn't see any difference between, for example, germ-free versus FPF at baseline. And we also placed these mice, placed the germ-free mice in a high fat, and we also observe a increase of ARVs expression in the skin in a mice placed in a high fat versus a short diet, for example. So the ARVs expression, for example, induced by the high fat, it doesn't depend on the microbiota like exposure, for example. So that's one point. So the germ-free in the skin, the ARVs expression in the skin germ-free is normal compared with, for example, SPF. But when you look at the ARVs expression in other tissues, for example, lung and the gut, the microbiota is important to trigger the baseline, the basal ARVs expression. For example, we have, for example, the germ-free mice has a lower expression of ARVs in a small intestine compared with SPF, for example. So that's one point. And when we associate germ-free mice with FMRP, we observe similar levels, similar ARVs expression levels compared with SPF. So the FMRP induced ARVs expression in SPF and also in germ-free. So in this case, there is no difference. So in general, just to summarize, what we have so far is that the ARVs expression germ-free, like the basal expression in the skin, is normal, but in other tissues, no, especially in the gut. It depends on the microbiota presence because the germ-free has less, in this case, less ARVs expression. But when we associate with skin convention, in this case, the FMRP, we didn't observe difference between the SPF versus germ-free. In the both situation, in both case, we have an increased ARVs expression in the skin. Yeah.
Djalma, we still have a couple of questions in the chat. The first one, Jan, do you think these results, as in the expression of retrotransposons, can constitute biomarkers in this context?
Yeah, in this, we can, of course, we need like more studies to affirm it, but in a context of, for example, steady state versus like a low grade inflammation, for example, that is induced in a high fat, what we found is exactly that. For example, the ARV's expression levels dictate homeostatic versus inflammatory response, for example. So if you have a discrete ARV's expression, we have homeostatic immunity. If you have aberrant ARV's expression, we have inflammatory process. So in this case, depend on the ARV's levels, we can say, like we can distinguish, for example, if the host is going to respond at homeostatic level or inflammatory level. So I think that we can say that, yeah.
Okay, and the last one I have here, someone with an iPhone asked you, in the context of a pathogenic bacterial colonization in which TLR2 and NF-kappa B would also be induced, do you believe that pathogens can negatively regulate endogenous retrovirus?
I guess that in the context of pathogens or infection disease or pathogenic bacteria, for example, we use a Staphylococcus aureus. That is a pathogenic bacteria, the skin pathogenic bacteria. And the Staphylococcus aureus induced much more than Staphylococcus epidermidis. So probably based just on this colonization and this association, for example, if you compare Staph aureus versus Staph IP, the Staph aureus induced much more ERVs than Staph IP. So based on this data, I think that the pathogenic bacteria can induce much more ERVs compared with non-pathogenic bacteria. And in the case of the other pathogens, I have also started some studies using Leishmania parasite. That is another kind of pathogen. It's a protozoan parasite. And the Leishmania infection, for example, in macrophages, is just a preliminary data, but we have a very high ERVs expression in macrophages after the Leishmania infection. So I guess that the pathogens, and it's an in vitro experiment, so we don't have other factors present in this case, just Leishmania and macrophage. So we can control better the experiment in this case. So we have a strong ERVs expression in this context. So I guess pathogens in general, for example, we have other reports showing that virus infection promote ERVs expression. Now I have one data showing that the helminths, I have some data showing that helminths infection also promote ERVs expression. And now recently we got that the Leishmania infection can also induce the ERVs. So I guess in general, the pathogens promote ERVs expression. So now we're going to try to understand what's the impact of this expression to the disease development.
Nice. Bruno, I'm guessing this question here you already asked, right? Is the same question, right? The one in the chat.
Sure. Thank you, Thiago.
Yeah. So any other questions or comments for Djalma? Anderson, Homer left you a greeting here also, Djalma. A lot of
great
people acknowledging. We got
thank you, Homer, for joining.
90 something people listening to your talk, and it will be available at YouTube for people to check it out later. So I thank you very much for being around, Djalma, and thank everybody to be here listening, and see you next week, everyone.
Okay,
bye-bye.
Say thank you again. Thank you, everyone.
Good job. See you.
Thank you.
See you.
Thank you, Djalma. Bye-bye.
Thank you.